So first of all, I would like to thank the organizers for giving me the opportunity to present part of our work here today. And I would like also to thank Manny for organizing on the French side of the iPoLS and bringing us here today. So the work I will be presenting today, we focus mostly on trying to understand the mechanisms and principles by which the bacterial chromosome is organized, how transcription and replication influences this organization, and particularly we focus also on the origin domain of the bacteria. So, to get you into a brief introduction, the bacterial chromosome is a circular piece of DNA that ranges sizes between 2 and 8 megabase pairs, and it has to be compacted about 1,000 times to fit into this cell here. And concerning the life cycle of the bacterial chromosome, the replication of the chromosome starts here at the origin and bidirectionally follows on, and while the DNA is still being replicated, the origins are very rapidly segregated to the nuclear poles, and they remain there while the rest of the DNA is being replicated. So the organization of the origin and the segregation relies on at least two kind of complexes. The first one is what is known as the partition system, that is composed of 3 elements: a repeat of DNA sequences that are called part S, a DNA binding protein called part B that will bind specifically to these part S sites and will create a complex that is known as the segrosome, equivalent of the kinetochore in eukaryotes. And then there is a third element, there is this protein called part A, that is an ATPase that will be responsible of interacting with the segrosome and segregating the origins. Now the second machinery that is involved here is known as the structural maintenance of the chromosome proteins, and I will name as SMCs many times. And they will be responsible for the resolution of the origin, and they are loaded into this sector here by the interaction with these complexes of part B, part S. So once the DNA is clear from the mid-cell, cell division takes place, and things go on. Previous studies that have been conducted in bacteria have the limitation of the size of the bacteria and the diffraction limit of light. So when we employ conventional microscopies and we try to look at the architecture of the DNA, we could not gather much information. So to overcome this limitation here, what we did is we employed a strain in which we label fluorescent-treated nuclear. For that, we take a histone-like protein from Bacillus subtilis, we label it with GFP, and in that way we can obtain images of the bacterial nuclear. We couple that with a super resolution technology that is called a structural illumination microscopy that allows us to obtain 3-dimensional images with resolutions of about 120 nanometers. So when we do this, the first thing that we observe when we compare a conventional image with a super resolution microscopy image is that we have these kind of elongated bundles that are connecting these regions of DNA that seem more concentrated. Now here I show you an image reconstructed in 3D of a bacterial nucleus in which you can observe this kind of regions that DNA is more condensed and that we term high-density regions. So the next thing that we do is, okay, we wonder if these regions, do they display like a subcellular specific localization, or does the localization of these regions varies with the cell cycle? So for that, we find the center of each of these, and the next thing that we do is we take cells of the same size and we accumulate their radial and longitudinal position, and we can build probability histograms of these high-density regions. So as you can see here, as soon as we increase the number of cells that we collect, very rapidly we observe here in black, there are certain high-density regions that display a very clear pattern. So now we can employ the metric as a metric of time, the size of the nucleus, and evaluate how these high-density regions are shifting their positions as a function of the cell cycle. So what you see here is how the nuclear size is changing and how the position of these high-density regions is changing. And what we observe is that for very small cells, we get a very well-defined high-density region in the middle that segregates very rapidly to the poles. It continues to shift to the poles as the cells get bigger, and then it gets back to the middle, and again the same cycle goes on. So as we can observe here, this matches very well with what I presented previously regarding the segregation of the origin domain. So the immediate question is, can we reveal the genetic identity of this high-density region? So for that, what we did is we combined the labeling of the nucleus with the imaging of the origin of segregation, of the origin, by labeling with the part B GFP, which is the protein I mentioned at the beginning. So when we do this, and we plot this kind of cosmograph in which you see the nuclear length, and here is the axial position of these high-density regions, or the axial position of this part B GFP protein, what we see is that there is a striking correlation between the segregation pattern of the origin and the high density region. So this clearly suggests that this high density region is the origin of replication. So once we did this, we have shown now that there is a new feature or architectural feature in the bacterial nucleus, and that one of those is particularly the origin and shows a very defined pattern. So the next question we wonder is, can we reveal more about the folding of the bacterial nucleus? So for that, we use a genome-wide technique that is known as chromosome conformation capture, that I will be very briefly introduce here. And by using this technique, what we get is a map of interactions between the genes with other genes. So the way this works is very simple. You take your bacteria, you just grow it, and then you chemically fix the DNA, and those DNAs that are in close interaction by proteins will be covalently fixed. So then you need just to digest this, a couple of steps, and you perform high throughput sequencing, and you get this kind of map here. These kind of heat maps that you see here have plotted in the Y and X axis the genomic position, and the color here in blue represents very low frequency of interaction, and what you see in white and red are high frequency of interaction between those loci. So to make this a bit more simple here, more intuitively, what you see is that when you have in the linear genome positions that are very close to each other, they will show a high tendency to interact. In that case, you will have this main diagonal that you see here that is highlighted in red. On the other hand, when you get genes that are farther away in the genome sequence and are interacting here, what you are moving is away from the main diagonal. Okay? So from this matrix here, we can obtain the topological representation of the Bacillus subtilis chromosome. So you see this kind of helicoidal shape that has been reported already for other bacteria, such as E. coli or Crescentus. And especially what is very interesting is that you can see here represented the interaction between the left and right arms that is described by this main diagonal that you see here. So the next thing that we can do is we can zoom a bit into this main diagonal here. And if you look at it, you will see that there are squares of different sizes. These squares here represent what we define as topological domains. So these are regions of DNA that have high frequency of interaction among themselves. And there is another feature that I would like to remark here, and is that some of these topological domains, if you zoom into them, you will see that you have a large square, and inside that square you have additional smaller squares. So those represent nested domains, in which you get domains that are interacting very frequently within themselves and with other neighboring domains. So this is kind of qualitative. So the next thing that you can do is you can employ something more statistically relevant, and there is different kinds of analysis. The one that we employ is known as the directional index analysis, and this will allow you to define topological domains. So here what you see is this matrix. You just take a piece, you rotate it 45 degrees, and the squares that you were seeing here now are these triangles that you are seeing here. This directional index analysis is what allows you to find these barriers defining these topological domains that I mentioned previously. And what you can do now is you can align the genomic positions of each barrier that you found and see how transcription is affecting the position of these barriers. And what we observe is that about 60% of highly transcribed genes that are represented here, so each peak here is representing the transcription profile of RNA, you see that 60% of them are colocalizing with these barriers. So this indicates that probably transcription, maybe by a supercoiling mechanism, is separating these domains. So what I would like now is to zoom a bit into the origin domain, that is this domain that I mentioned previously, that was very interesting for us. So when we zoom here, the first thing that you notice is that we have this main diagonal that I described previously, and we naively thought that maybe this diagonal was representing this left-right interaction between the two arms of the chromosome, and that the tip of this interaction was given by the origin of replication. But when we look at the position of the diagonal and where the origin is here in violet, what we observe is that this is not the case, and actually the diagonal is traversing 100 kb to the left side of the arm. Now, this represents the hairpin of this kind here that we call primary hairpin. Now, you may notice that there was a second diagonal in the first image I showed that is here. And this diagonal here that is about 350 kb to the left represents an intra-arm hairpin or a secondary hairpin. So this is giving us a first picture of the folding of the origin domain in which we have these two kinds of hairpins. So the immediate question is, who are the molecular actors responsible for the formation of this structure, this folding here. So what you can do is, now we are going to zoom a bit more here, and when we do that, we will align our matrix here with these protein enrichment profiles. So what you're seeing here are the genomic positions in the x-axis, and each peak that you see here represents enrichment of a given protein. So for this case, we are just showing the SMC protein, DNA protein, which is a protein that inhibits replication, and par B, that is the protein I mentioned at the beginning. And here, on top of this, I'm showing you the par S that I mentioned, that are these sequences to which par B is binding. So when you zoom a bit more here, what you find is that the tip of this primary hairpin that I described previously is highly enriched in this par B protein, and also is very enriched in these SMC proteins that also are binding and diffusing from this position here. Now, if we now zoom at the secondary hairpin that I mentioned previously, there is something that is very interesting. You see these red dots here, they represent a specific interaction. And you can look at the position of this specific interaction, and it's matching the position of the parasite with the par B enrichment protein that you see here. And very interestingly, it's bringing together two distant arms and bringing together those two DNA boxes here that are those inhibitors of replication that I mentioned, and they are kind of gathered together by these SMC proteins, so they do this kind of ring. So if you continue to play this game around, you can kind of like build a puzzle in which you can do a schematic prediction of the folding of the origin domain. So we build the puzzle here, and the next thing that we wonder is, we want to make sure that actually what we are saying, that these proteins are really responsible for this. So we built strains in which we took out sequentially each of these factors here. So when you take away SMCs, par B or par S, what you see actually is that the main diagonal that I mentioned previously is completely gone. So this is indicating that the interaction between the left and right arms is given by the presence of these components here. And not only that, when you look at the full matrix, so here you see the matrix going through the whole chromosome, the main diagonal is completely gone. So this is not only affecting the folding of the origin domain, but it's also affecting the folding of the whole chromosome. And here you get again a topological representation in which you see now the chromosome completely open. So the next thing that we wanted to verify is, okay, can we now look this at the single cell level and live cells? So for that, we took the origin of replication, and we found those two positions that were very adequate for what we wanted to test as a hypothesis. And we label with these fluorescent positions here, and we test the effect of removing this par B protein, or when you are in the wild-type case. And what you see clearly is that the presence of this par B protein, that is also loaded these SMC proteins, is leading to a decondensation of the origin domain. So the next thing is, I mentioned at the beginning, we were also interested in seeing how replication is affecting the folding of the origin domain. So for this, we can take advantage of a particular strain of Bacillus subtilis in which you can inhibit replication. So in this matrix here, replication has been inhibited. So replication will continue until there, but a new cycle will not be launched. And next, you can just change the temperature in which you're growing your cells, and they will restart replicating. So the first thing that you observe is that the main diagonal is completely gone, and that as replication takes place, the diagonal starts to reappear here. If these matrices look a bit more noisy than before, it's because the number of reads are lower. So what this represents here is that when you're in this situation in which replication is arrested, probably you're facing an open origin domain, is represented here. If you look, there is still a bit of the secondary diagram that is in our scheme we are representing here, and this matches with a chromosome in this kind of configuration. Now, as replication takes place and everything restarts, what you see is that the origin domain is becoming refolded, and these DNA boxes are becoming close back together. And we come back to this representation in which you have a longitudinal representation distribution of the chromosome. So this clearly is telling us that replication and segregation is playing a role in the folding of the origin domain. So what we wanted to do next is, can we look at the folding of the origin by super resolution microscopy? And for that, we took advantage of this par B protein, and as I mentioned, these parasites are distributed along the origin domain. And when we label this and we perform structural illumination microscopy, we observe that there are subclusters of par B that you can see here. So we use the same strategy as before for the whole nuclear labeling, and we can find the maxima of each of these subclusters here. and we can measure the size of the origin domain as the maximum distance between each of these subclusters. Now, just to make sure that this was not an artifact coming up from the 3D SIM experiments, we performed the same kind or similar kind of experiments, but now employing 3D PAL, in which we can get resolutions of about 40 or 50 nanometers, and still you can see, you can observe very clearly these subclusters here. So, as previously we have the origin domain sizes, and we have thousands of cells, so we can classify this according to the cell size and recover kind of a cell cycle in this kymograph that you see here. So again, you get the nuclear length here, the axial position, and the color code here is telling you about the size of the origin domain. And what we see is that the origin domain, when it's in the middle of the cells, is much larger, so it's much decompacted than when it's in the poles. And what you see here is the branches when the origin is being replicated once again and migrating to the poles. So, and here, what you see is the number of subclusters, so the behavior is pretty much the same. That's pretty evident. So, to finish with this, what I would like to present to you is what we think is happening in Bacillus subtilis, is that actually you have these chromosomal conformations coexisting, in which probably in rapidly growing cells, this is the one that is predominant, and that the replication will trigger a defolding of the origin domain, and trigger also this segregation pattern that I mentioned previously, and that the recipient of the chromosome will rely on a mechanism led by the SMC proteins, in which they will load at these parent sites here, and they will form a closed ring and maybe diffuse into the terminus, and probably by stochastically or nucleotide-related mechanism, open up and to come back to this situation over here. So, with this, I would like to acknowledge all the people that is part of this work. I would like to acknowledge Marcelo Norman, who is our group leader, who has guided us in this work. Antoine, who is a postdoc at the lab, and he has done most of the microscope experiments with me, and he developed all the analysis tools that allow us to obtain all this data. Our collaborators from Paris, they have done all the high-C measurements and analysis of data, and our collaborators from Newcastle that are the ones that help us with the build the strains and the genetic studies. And I thank you for your attention.
Thanks, that was very interesting. I just wonder if you could comment on the, how you go from these matrix maps to structural information. Is that a unique problem? I mean, how much confidence can you have in the structure?
Yeah, well, that structure is a topological representation, so there is no minimization of energy or anything. It's just to give a picture of the chromosome. So it doesn't have a physical meaning in the sense that there are no true distances there. So our goal in the future is trying to combine microscopy imaging with this in order to build a true chromosome. So that's just topological.
That was just really beautiful. I have several questions, but I'll ask only one. It seems like there's a pretty steady time to replicate the entire DNA, circular DNA in E. coli. It's about 40, maybe 45. Do you understand that?
No.
Can you give us? Oh.
You have many questions. So
okay, I'll move on to question 2 then. And then the question 2 is: have you looked and compared your work to this nice paper that Sunny Shi did, where he labeled HNS, and looked and also compared it with chromosome capture data?
Yeah, I know which paper. No, we haven't compared that one. I know which paper you're talking about, but no, we haven't compared that one. So.
This was more detailed.
Yeah, no, we haven't contrasted the work with this. No, but I know which paper you're talking about.
Do you see a qualitative difference between fast and slow growth rates when you get to the multiple origins regimes?
No, we haven't seen huge differences. Differences are minimal. And actually, when you look at the transcription profiles, differences are not so big either way. So we have done this in rich media and minimal media, and we get pretty much the same results. I have the matrices for the other ones, they're pretty much the same.
And what you showed today, which growth rate was that?
Everything was minimal media here. It's like much nicer, especially for microscopy. For matrices, it's nicer with the rich linear. Right.
Okay, thank you, Diego. We're gonna move.